All right, welcome back. In this lecture video, we're going to transition from looking at the design structure of an experimental design to the treatment structure of an experimental design. And in particular, we're going to be focusing on complete-factorial treatment structures. So let's take a look at an introduction here. Up until this point in the course, we've been focusing on the design structure of an experiment. Design structures pertain to how an experiment is actually conducted, in particular, how treatments are randomly assigned to the experimental units used in an experiment. The manner in which treatments are randomly assigned to the experimental units determines the design structure of the experiment. The designs we've looked at up to this point include the completely randomized design, the randomized complete block design, and the Latin square design. Now, if you think about it, the primary thing that differentiates these three designs is how the treatments are randomly assigned to the experimental units. In the completely randomized design, randomization is completely unrestricted. That's why we call it the completely randomized design. In the randomized complete block design, each treatment is randomly assigned to one experimental unit within each block, but each block receives its own separate randomization. And finally, in the Latin square design, treatments are randomized to the experimental units that make up that Latin square, subject to the constraint that each treatment occurs only once in each row and only once in each column. Now, in terms of the randomization processes for these three designs, the Latin square design is more restrictive than the randomized complete block design, and the randomized complete block design is more restrictive than the completely randomized design. Now, identifying the correct design structure used in an experiment is crucial because in order to make valid inferences from statistical analysis of data generated by the experiment, the design portion of the statistical model must conform to the design structure that was used in the experiment. The design structure used in the experiment determines the appropriate design structure components of the statistical model that must be used for results to be valid. Even if some of the components of the design structure part of the model, such as one or more blocking factors, are not statistically significant, it is not appropriate to remove insignificant design components from the model, because doing so changes the model from the one for the experiment that was actually conducted to a model for an experiment that was not performed. So it is not appropriate to analyze data from a Latin square design with a model for a randomized complete block design. Likewise, it's not appropriate to analyze data from a randomized complete block design using a model corresponding to a completely randomized design. But another important aspect of the experimental design is the set of treatments used in the experiment. The structure of the set of treatments used in an experiment is called the treatment structure of that experimental design. Since the goal of experimentation is to assess, evaluate, and compare the effects of treatments on one or more response variables, selecting the set of treatments to be used in an experiment is often one of the first things, if not the first thing, that is done by a researcher in the experimental design process. Careful selection of treatments is required in order to ensure that all research questions of interest can be answered once the experiment has concluded. In addition, careful selection of a treatment structure is also necessary. It's also required to ensure that questions that must be answered as part of the statistical analysis itself can be answered. Now, what am I talking about there? Well, in performing a statistical analysis, there are various steps that one has to go through, and the steps that one takes often depend on testing whether certain assumptions about the model are appropriate. All right, and so even if these questions are not part of the set of research questions for the researcher, there are questions that need to be answered in order to do a proper statistical analysis. All right, and so we need to make sure that the treatment structure is such that those questions can be answered as well. Finally, careful selection of the treatment structure can help ensure that the experiment is as efficient as possible, in the sense of providing as much information as possible about the effects of the treatments on response variables relative to the resources expended in conducting the experiment. All right, a researcher wants to get as much bang for the buck from the experiment that they're conducting, all right, for the money and time that they're investing in the experiment. They want to get as much information as possible. And that can be facilitated by ensuring that an appropriate treatment structure is used. Now, most experiments are performed in the context of a variety of conditions that are under the control of the researcher. They're under the control of the one doing the experiment. And these conditions are often conditions that can affect the response. Now, even in situations where there is only one factor of primary interest to a researcher, the experimenter must make decisions regarding a number of other factors that make up the conditions under which the experiment will be conducted. And these other factors will almost assuredly impact the response. In order to understand which factors affect a particular response and the nature and magnitude of such effects, it is necessary to vary the experimental conditions that experimental material is exposed to by varying the various factors that can affect the response and measure the response under these varying conditions. Now, the traditional approach to experimentation has been to vary one factor at a time. While this might seem like a good idea from an intuitive standpoint, the one factor at a time approach is actually what one does not want to do in experimentation. Now, why is that? The one factor at a time approach suffers from the following problems. First, it's an inefficient use of time and experimental resources. And we're going to talk about these more as we go along, but that's one of the reasons why this is problematic. All right, it's an inefficient use of experimental resources. Two, it's based on faulty a priori assumptions about the way in which the factors affect the response. Third, the one factor at a time approach can produce misleading results, certainly incomplete results, and often because of that lack of information, a lack of a complete picture of what's going on, the results drawn from one at a time approach experimentation can actually lead to misleading results. And finally, the one factor at a time approach cannot be relied upon to find conditions that lead to optimal values of the response. So the one factor at a time approach is not preferable. In fact, that's exactly what we don't want to do. So what's the alternative? Well, a much better approach to experimentation, especially when trying to identify factors that affect the response, is to cast a much wider net, so to say, by flooding the predictor space with a net of conditions to which experimental material is to be exposed in the experiment and the response measured under those various conditions. Now, this is accomplished by simultaneously varying the factors under the control of the experimenter and doing this in a systematic way. This approach offers several advantages over the one factor at a time approach. First, no unwarranted assumption is required, no unwarranted assumption is made regarding additivity of the factors in their effects on the response. A complete factorial treatment structure allows for non-additivity of the factors. In addition, the additivity assumption can actually be formally tested, and further analysis can proceed based on the results of that test. When the assumption of additivity is not rejected, this approach provides equal replication for estimating the main effects of all factors under consideration. Let me say that again. And the reason this is important is because this is one way, a big way, in which this approach is much more efficient. It's a much more efficient use of resources than the one factor at a time approach. Because when the assumption of additivity is met, when it's not rejected, all right, then this approach that we're going to be looking at, the complete factorial treatment structure, provides equal replication for estimating the main effects of all the factors that are being considered in the experiment. So that's a really big advantage to this complete factorial treatment structure approach. Now, what if the assumption of additivity is not appropriate? What if it is rejected in that formal test? Well, if the test of the assumption of additivity is rejected, then this type of treatment structure provides response measurements under a large enough variety of experimental conditions for studying the ways in which the various factors may in fact interact with each other in their effects on the response. This is a very important aspect of this approach, since interactions among factors is common, and because the one factor at a time approach provides a set of experimental conditions that is not broad enough. It's too narrow to allow full investigation of such interactions. So that's the intro to this particular module on complete factorial treatment structures. In the next lecture video, we'll talk about what a complete factorial treatment structure actually is. So what is a complete factorial treatment structure? A complete factorial treatment structure is a set of treatments consisting of all possible combinations of two or more factors, each at two or more levels. Each level of every factor occurs together in combination with each level of every other factor. Such a set of treatments is highly structured, and that structure serves to motivate and provide the necessary data for making many important and relevant comparisons among the various factors that make up the treatment structure. Now consider a factorial treatment structure generated by k factors. Let's suppose that factor 1 has n1 levels, factor 2 has n2 levels, factor 3 has n3 levels, and so forth down to the last factor, factor k, which has a number of levels represented by n sub k. Then, by the fundamental rule of counting, the number of treatment combinations comprising the complete factorial treatment structure generated by these k factors is equal to the product of those numbers of levels. So n1 times n2 times n3 and so forth up to times n sub k. Now, because of this, because the number of factors grows multiplicatively with the number of treatment combinations, we can see here that the number of treatment combinations increases very quickly as the number of factors increase and as the number of factor levels within a given factor level increases. So as an example, suppose that a factorial treatment structure consists of two factors, which we'll label A and B. Let's further suppose that factor A consists of two levels, which we'll denote by A1 and A2. Factor B has three levels, which we'll denote by B1, B2, and B3. So with this setup in this context, there are 2 times 3 or 6 treatment combinations making up this complete factorial treatment structure. Now the 6 treatment combinations are as follows: A1, B1, A2, B2, A1, B2, A2, B2, A1, B3, A2, B3. Now, since each level of factor A occurs with each level of factor B, this is a complete factorial treatment structure. As another simple example, suppose that a factorial treatment structure consists of three factors labeled A, B, and C, where in this situation factor A has three levels, denoted by A1, A2, and A3. Factor B has three levels, denoted B1, B2, and B3. And factor C has four levels, denoted by C1, C2, C3, and C4. In this situation, then, there are 3 times 3 times 4, or 36 treatment combinations making up this complete factorial treatment structure. Now, as an exercise, list the 36 treatment combinations for this complete factorial treatment structure. All right, next let's look at the nature of the impact of the factors on the response in a complete factorial treatment structure. So additivity versus non-additivity. We've used this term, but we haven't defined it yet, so let's talk about what additivity means. In a factorial treatment structure, where the levels of two or more factors appear in combination, the factors can affect the response in a variety of ways. If two or more factors affect the response independently of one another, then the effects of those factors are said to be additive. Okay, let me say that again. The effects of two factors are additive if their effects on the response, if they affect the response independently of each other. Their combined effect on the response, in this case, is the sum of their individual effects. On the other hand, it's often the case that the effect of one factor depends on the levels of two or more other factors. In this case, the effects are non-additive because their combined effect on the response is different than the sum of their individual effects. In this case, the levels of the factors are said to interact. Another way to say this is that an interaction exists among the levels of factors whose effects are not additive. So what are the advantages, disadvantages, and uses of a complete factorial treatment structure? When the factors affect the response additively, that is, when there is no interaction of the factors with respect to their effect on the response, we have several advantages. First, there is a simplified interpretation of the factors. Main effects are all that are needed to describe how the factors affect the response variable. All right, that's going to be the case when there is no interaction between the factors. Right, when the factors act on or impact the response additively. In addition, and this is this big advantage when it comes to increasing the efficiency of an experiment. When there is no interaction, when the factors impact the response additively, then there is hidden replication. All right, and what this means is that each main effect, the main effect of each factor, is estimated with the same precision as if the entire experiment had been devoted to that one factor alone. And this is one of the big reasons why a factorial treatment structure, an experiment with it using a factorial treatment structure, is much, much, much preferred over an experiment that uses a one factor at a time approach, because the one factor at a time approach does not have this characteristic. There is no hidden replication there. All right, but with a factorial treatment structure, there is this hidden replication when the factors impact the response in an additive way. In other words, when there is no interaction. Now, In many cases, there will be interaction. All right. And the problem with experiments that are done using the one factor at a time approach is that uh, experiments done in that way do not provide the data to fully understand what's causing the non-additivity. You can't fully understand. You may not even be able to detect that there is an interaction. But when using a factorial treatment structure, when there is interaction, the complete factorial treatment structure provides the data necessary to estimate all the interactions among all the factors and to estimate them all with equal precision. So that's another big advantage, another reason to use a factorial treatment structure as opposed to the one factor at a time approach. Now, what are some disadvantages? Should we call these disadvantages or just characteristics, all right, of this approach? But as we've seen in some of our examples, the number of treatment combinations increases very quickly. It increases multiplicatively with the number of factors used, so that the size of the experiment, the number of experimental units that are required for the experiment, increases rapidly as the number of factors increases. It also increases for a fixed number of factors. The size of the experiment increases with the number of levels that one or more of those factors have. When interactions among factors exist, factorial experiments with a large number of factors can be difficult to interpret properly. Now, that's certainly true if you've got 8 factors in an experiment, which is not necessarily that unusual, especially in situations where experimenters, researchers are trying to identify which factors are impacting the response, doing it in, for example, a preliminary study, some kind of a pilot study. And once they get the data from that kind of an experiment and they determine which of the factors are important, they can do subsequent experiments using those factors. But in a situation, in an experiment, using a factorial treatment structure with even 4 factors, if interactions do exist, it can be difficult to properly interpret what those interactions mean. Now, is it really fair to call that a disadvantage of the factorial treatment structure? Or is it? I guess one thing I would say about that is that it's just the nature of that response and the nature of how the various factors that impact the response can influence each other in their effect on the response. So it's really a reflection of the reality of the situation in nature. And whether we want to call this a disadvantage of the factorial treatment structure or an advantage, one could argue that by virtue of the factorial treatment structure illuminating these interactions, this non-additivity, that's actually an advantage of the factorial treatment structure. But do keep in mind that as you increase the number of factors, the experiment does get bigger, and when interactions exist, you'll need to deal with those. And then finally, what are some uses of the complete factorial treatment structure? Well, they're very useful in exploratory experiments like I was just talking about, for examining a large number of factors to determine which factors are important, which factors are impacting the response, and therefore should be studied further, and distinguishing those from which are not important and really don't need to be included in future experiments. It's also useful to investigate the relationships among factors with respect to how they affect the response. That is, do factors affect the response additively, or do they affect the response non-additively? That is, are they independent in their impact on the response, or are there one or more interactions among the factors? And then finally, they're useful in experiments whose results will lead to recommendations over a wide variety of conditions. the most important or most prevalent conditions can be included as factors in the experiment, even though they are not the primary factors of interest. So, for example, let's suppose let's think about an agricultural experiment, all right, and the results of the experiment are going to impact how farmers conduct their field work, all right, how they plant, harvest, and whatever. Well, farmers in different situations, different locales, they may have different types of soil. They may have different levels of. They may have irrigation or non-irrigation. They may have different levels of natural moisture in the soil. The conditions that different farmers work in they're going to vary from one farm to another. And even though those kind of conditions may not be the primary factors of interest to a researcher, the researcher can incorporate these kinds of factors in their experiment so that when farmers, for example, go to apply these results, they can apply them to conditions that more closely match their own environment. So those are some of the uses of the complete factorial treatment structure.